Good afternoon, children. Yes, what is going on? Yes, good afternoon, everybody. Still on lunch? So, how are you? How is cold over there? Good afternoon. So, where are your friends? Okay, just inform them we have started the class. Enough of sunbasking. Okay, fine. So, did you revise the last chapter that we completed yesterday? Did you revise it? Did you go through that? Yes or no? Yes. Yes. Yes, Tamil Nadu, Shillong, Itanagar. Everybody. You can't hear me? Is it? Just a minute. Okay. Am I audible now? Yes, Shillong. Okay, so we are going to start a new chapter today. Can you hear me now, Shillong? Am I audible? Okay, great. So, we are going to start a new chapter. I guess we don't have any doubts in the last chapter. In case you have, you can always discuss. That's always, you know, you people are actually not asking questions. I don't know why. But please start studying because we have done many chapters. We have completed a very big bulk of syllabus in biology, and yes, you know there are 100 questions going to be asked, 90 questions going to be asked in your NEET exam from only biology, isn't it? So we are going to start a new chapter, that is microbes in human welfare. Okay, this is a good chapter. You need to only learn things. This is a very good chapter. So let's see how good you are at mugging up things. So it's a chapter with lots of names of different types of microbes which are obviously useful to us. So last chapter was basically about those organisms or those microorganisms in like bacteria, protozoa, fungi. So those microorganisms which were harmful for humans, isn't it? And this chapter is about human welfare. So human welfare is what? What is human welfare? So anything which is basically, we understand that the microbes are very important components, isn't it? Apart from being a nuisance, they are important components. In fact, we do have microorganisms in our system also called as good bacteria, friendly bacteria, and they help in absorption of many important vitamins, isn't it? So microorganisms are important components and you can find them everywhere. That is in soil, water, you name the thing and you will find the microorganism inside our bodies. Like I told you, the good bacteria, and inside plants and other animals also. So you're going to find these microorganisms almost everywhere. So this almost everywhere is not only limited to soil, water, air and all those things, but under harsh conditions also. So Vineet sir must have taught you that they are present deep inside the geysers. You know what is a geyser? That is thermal vent. And these geysers have a temperature of more than 100 degrees Celsius. So it's a very high temperature. 100 degrees Celsius is a very, very high temperature, and you can find them over there also. Not only this, but you will find them deep inside the soil, layers of snow, highly acidic environment. So I must have told you, if it is found under layers of snow, the particular bacteria is called as psychrophile. P-S-C-H-Y-R-O-P-H-I-L-E. And if it is under acidic condition, it is said to be acidophile. And so if it is found inside the geysers at a very high temperature, they are said to be, I will be writing over here, thermophile. So thermo means temperature and phile means loving. So they are loving high temperature, isn't it? You know, Thermus aquaticus is a thermophilic bacteria, and we use that bacteria to isolate a particular DNA polymerase enzyme, which is very, very useful in PCR. The DNA polymerase is called as Stack polymerase. So still the strength is very low at Shillong. Yes. Why? What's the reason? Still issues are there. Many of you have not turned up for the class. Is everything okay at Shillong? Yes, Tamilong? Is everything clear? Itanagar, Nabam and Nuli, Raleigh, okay, curfew, still curfew is there. Oh, sad. Okay. Sad. Okay, let's see, let's move ahead. So these are the particular things you need to understand about microorganisms. So what are microbes? Let's classify microbes. Is Nabam, Luni, is it clear? So what are microbes? We know microbes as protozoa, bacteria, fungi, microscopic animals, we have plant viruses, we have viroids, and we have prions. So I must have taught you viroids and prions. So we have these particular kind of organisms which are placed under the term microbes. So microbes is a very, very general term, and we can see these many organisms placed under only term microbes, isn't it? But what we are interested over here is only two microbes. Actually we will be dealing in, so we are interested more in bacteria and fungi for human welfare. Why? Because they can be grown on nutritive media. So if they are able to grow on nutritive media, we are basically interested in that, because what we need their products, and their products will only be isolated, can only be isolated only when we can grow them at our will. So the nutritive media, they support the growth of the colonies, and these colonies, yes, you can see them, can be seen by naked eyes. So these colonies, they are useful in studies on microorganisms. So if you wish to study microorganisms, you have to keep in mind that you need what? You need colonies, and those colonies can be grown on nutritive media, isn't it? So that actually gives the conclusion that when we talk about microbes, they are not always harmful. But sometimes are useful also. Let's see their uses. So we are talking about two important microorganisms here, that is bacteria and fungi. So if you wish to study any microorganism, you need, the first priority should be that it should be able to grow on the nutritive media. So you should be able to grow them at your own will. That would be the first and foremost requirement. If it is not, it would not be very useful for us, because at one point of time the source is going to get extinct, it is going to get over, isn't it? Copy it? Did you copy up to here? Okay, very good. Now let's talk about microbes in household products. The first thing. So how many of you have heard the term curd? Have you heard about curd? Nabam, Loni, Shillong? Have you heard curd? Yes. What is curd? No, you did not? Ravilong? Have you heard about curd? Itanagar? Yes. Spoiled milk is not curd. Spoiled milk has to be thrown. You cannot consume spoiled milk. If it has spoiled, you cannot consume it. But fermented, not exactly fermented, but yes, we can talk about, like, we culture some sort of bacteria called as lactobacillus. Isn't it? So lactobacillus and other lactic acid bacteria. The C/A is called as LAB. This grows in the milk. This is going to grow in the milk and convert it into curd. So lactic acid bacteria that grows in the milk and converts it into curd, and this actually, how does it happen? This LAB or lactic acid bacteria, this produces some acids. That coagulate and partially digest the milk proteins. That is why if somebody is lactose intolerant, somebody is not able to digest milk, they are advised by the doctors to have curd. Why? Because already the lactic acid bacteria has digested the parts of the milk protein partially, that is to an extent. Now, what we do is that this lactic acid bacteria, a small amount of curd, when added to fresh milk, it leads to multiplication. Of bacteria. So, what we do is that we take a small amount of inoculum, that is called as inoculum, and that inoculum is put into slightly warm milk, okay, and that particular inoculum starts to grow overnight into that whole milk component that has been, to which it has been added, and gradually the bacteria starts to multiply in the milk, particular milk that you have left for overnight. Now, gradually they produce acids, and those acids they coagulate the milk proteins and start to digest it partially. The milk proteins are getting digested, and this is not only helpful in the digestion process, but yes, this also increases the most important thing, vitamin B12 content of the milk. So, this is a very typical thing, that is, it increases the vitamin B12 component or vitamin B12 content of the milk. So, this is a very good product which actually is helpful. Like if you talk about its good effects, this lactic acid bacteria checks disease causing microorganisms. In our stomach. So, in a way it controls that how much of disease causing microorganisms should be present, and if they are present, what is this going to do? This is going to destroy them. So, it is very useful. You can see as such that I have highlighted three things, that is, first of all, it is helpful in making it more digested, more partially digested, then it increases the vitamin B12 content, and then it removes out all the harmful bacteria. So, in this way the consuming curd is thought to be very healthy lifestyle. Okay. So, do you consume curd on a daily basis? Not curd, I guess, lassi. Lassi is very popular. No lassi, no curd. Okay. Why? Don't like the taste? Huh? Don't like the taste? Lassi or curd? No. Okay. You like it? Lassi? Because my class 12th students, they were very excited with lassi. They were like, okay, what is going on? What are you searching for? What is it? Yes. So, when I was teaching this chapter in class 12, they were very excited with the lassi word. So, they were like, okay, they could not relate it with curd, but when I told them lassi, they were very easily able to relate it. Okay, we get to have lassi over here. It's packed in bottles, and that we get to have. Shillong children only. Fine. Yes, Tamilong. Itanagar, where is Looney? Yes, Itanagar, are we connected? Yes, Nabam, is it sorted? Clear? Just let me know if it's you have copied this. I guess we have copied, isn't it? That's why we are busy in chit chat. Okay, fine. Okay. Next thing is, yes, Lunia, are you able to understand? Nabam? B. So this was A, curd. Okay, fine. So this B is next thing what we get to have with the help of these microorganisms. So we have dough of dosa and idli. So this is also fermented, isn't it? This is also fermented product by bacteria. So do you consume this? Okay. No problem, Nooni, no issues. Just let me know in case you don't get anything. You can talk to me on group also. So dough is basically, why it is important? Because this gives a puffed appearance. Is due to production of carbon dioxide gas, isn't it? So when there is particular process of fermentation, we know there will be production of CO2 and ethanol, isn't it? Respiration, remember there is a chapter in respiration in plant physiology where sir will be teaching you, or sir might have taught you two things. If it is like yeast or any kind of bacteria undergoing a process of anaerobic respiration, they produce two things. Number one, they produce CO2, number two, they produce ethanol, isn't it? And both of them are very, very important economically for us. So this is basically why this puffed appearance is basically because of CO2, and this is fermented by using baker's yeast called Saccharomyces cerevisiae, isn't it? So commercially if you go and have this, that will be used by baker's yeast, isn't that? It's called as Saccharomyces cerevisiae. But many times we ferment it using bacteria also. Normally if you just prepare the dough and leave it overnight, it will itself start to produce fermentation process, and that puffed appearance will appear without adding baker's yeast. But yes, if you are using it commercially, you need to put some baker's yeast in order to have more of fermentation or faster fermentation. Now you have traditional drinks also. This would be under C, like toddy. This toddy is from South India. So it's a kind of fermented local drink which is produced by fermenting sap from palm. So sap is the particular product that comes out from the palm trees or any kind of plant, that white milky production or watery part. What is going on at Shillong? What is it? Huh? Why are you not paying attention? Everybody? This is a very easy chapter, that's why, is it? Please don't talk. This is not very easy chapter. You can have questions from this chapter as well. Don't take any chapter very lightly. Questions from each chapter, if they are coming, they will be coming of 4 marks. Whether it is from a difficult chapter or an easy chapter. Okay. Okay, fine. Okay, so this is a traditional drink from South India. So did you have this? Balajit, where did you have this? But I guess you are under 18. Are you? Isn't it? Are you 18 plus, Balajit? You should not consume alcohol or any kind of local drinks and all those things until you are 18 plus. Isn't it? Okay, fine. So this is okay, fine. So he is Khasi. Khasi is a particular group of, I guess, part of Khasian hills, isn't it? As far as I know about northeast, it's a particular part of Meghalaya, is it? Just correct me if I am wrong. Part of hills. Okay, fine, Balajit. So you are able to connect it very nicely. I know you will be must be happy with this. So traditional drink like toddy. So every state has a traditional drink of its own. Your NCERT just placed this drink in your book. That's the only thing. Okay, moving ahead. So what we have is, so please don't try these things unless and until you are 18 plus. So I won't be encouraging these things. Once you are 18 plus, you can do anything. Not exactly anything, but yes, these type of things. So microbes ferment not only these things, but yes, they are known to ferment fish, soybean and bamboo shoots also. So this must be, I guess, very much relatable to you, because northeast this is very rampant. Fermented soybean, fermented bamboo shoots, they are very part of all, I guess, all the cuisines, isn't it? Is it? Fine. So we'll move ahead. Yes, Tamilong, Itanagar, just let me know in case you have any doubts. This would be the D part. Household products, 4 things we did see. So next thing is cheese. So what is cheese? Say cheese. So cheese is what we have, different varieties of cheese. Isn't it? And these different varieties are different because of their different texture, their different flavor and their different taste. So because of all differences, because of all things which are coming in, like falling at different things, what we have is, we have different varieties of cheese. You like cheese? Boys, I know they like cheese very much. Yes? No? Okay. So cheese are different. Why cheese are different? Why cheese are going to get different? Because of the microorganisms we use to ferment them. Cheese are different because of characteristic microbes used in their fermentation. We will see some examples. So we know some cheese, you must have seen that, especially in Tom and Jerry, Swiss cheese. Now this Swiss cheese is known for its large holes, and this is why, because of production of carbon dioxide by a bacteria called as Propionibacterium shermani. So this Propionibacterium shermani is a particular species. This S would be always small, keep in mind. So this is biological nomenclature, and this produces CO2, and that CO2 is responsible for very large holes in the Swiss cheese. So this is how you can make Swiss cheese. You need to have this particular microorganism for the same. Next we have is one more example of from cheese, Roquefort cheese. So this Roquefort cheese is again having a different recipe. This is ripened by growing a specific fungi on them, and because of this specific fungi that gives them the particular flavor. So we have this particular thing, two things we did see now. What are they? Number one is Swiss cheese and number two is Roquefort cheese. So this whole cheese business is basically because of different kinds of microorganisms that we use to make that. So different flavor, texture, appearance, everything is differing because of the type of the microorganisms we are using on them. Yes, are we clear? Everybody, Itanagar, Tamilong, Shillong, just give me a thumbs up if you have copied everything. Tamilong, where are you? Good. So next time you are having a cheese, don't forget to get to know that what kind of microorganism has been used for its fermentation. Now, next section would be, we did microbes in household products. Now we will talk about microbes in industrial products. This would be the next sections. So we have next topic, that is microbes in industrial products. So industrial products, how microorganisms are helpful for us. Let's see. So these microorganisms, they are known to synthesize a lot of products. Synthesize a number of products. For example, if you like just move around and see, notice you have beverages, you have antibiotics. They are the most important things which are actually produced by microorganisms. So what is the requirement? The requirement basically is we need to produce these things on a large scale. and this requires scaling up, and this requires large vessels called as fermenters. So we have large vessels, we do have biotechnology part when sir will be teaching you applications and all those downstream processing section. He will be telling you, he will be showing you two types of bioreactors. One would be the stirred tank reactor and one would be the sparge tank reactors. So those reactors, those biofermenters are very, very important when you need to scale up your products, the particular things which are produced by microorganisms using them. So we will start with fermented beverages. So we have beverages of wine, beer, whiskey, brandy or rum. So we are aware that these last three, that is whiskey, brandy or rum, we must be aware that they are distilled, but wine and beer are only fermented. So once you produce the fermented product and if you go for its distillation, its alcohol content actually increases to a very high level. So out of these two, if a question comes to you, you have to mention that they have comparatively a lower alcohol content, isn't it? The content of alcohol will be very, very low in wine and beer compared to whiskey, brandy or rum. Why? Because these are distilled products. They are produced by distillation. Yes. What is it? Copy this. Yes. You have lots of time to chat. Copy this, please. All these fermentation, all these products are fermented by a yeast called as Saccharomyces cerevisiae, and this same Saccharomyces cerevisiae is used in bread making. The other name for Saccharomyces cerevisiae is, if you use it for bread making, it would be called as baker's yeast, or if you use it for fermentation of beverages, it would be called as brewer's yeast. You know the reason why. If ethanol is produced, it would be brewing, that is the particular product of concentration of alcohol would be increased, and if you are talking about baker's yeast, what we need is, we need puffiness. So your cakes, isn't it, your cakes which you prepare at your home, that won't be that fluffy if you don't use the baker's yeast. Have you seen that? Yes, Tamilong, Itanagar, Shillong. Have you seen baker's yeast? Yes or no? Have you seen your mother using those yeast for preparing cake dough? You must have. Have you? Yes, Christmas is on the corner. Yes, fine. Okay. So we do have these kinds of products which are basically very, very helpful, and we need some sort of microorganisms to help us prepare these tasty things, isn't it? I'm talking about cakes. Okay, so what is brewer's yeast doing? So baker's yeast is doing this thing, isn't it? The baker's yeast is doing bread making. And what is brewer's yeast or same Saccharomyces cerevisiae? What is it doing? This is doing what? This is fermenting. This is performing fermentation of malted cereals and fruit juices. To produce ethanol, isn't it? And ethanol is what? Ethanol is, so can you consume ethanol directly, or you need to process it? So we have done fermented beverages as the first product of industrial usefulness, that is fermented beverages. The next product would be let's say antibiotics. What are antibiotics? Antibiotics are the chemical substances which are produced by some microbes and can kill or retard the growth of other disease causing microbes. So this whole idea was like first antibiotic. It was China's discovery. It was penicillin. So first antibiotic to be isolated, to be discovered was penicillin, and this was purely a chance discovery. Do you know the how this was discovered? Penicillin story. Do you know about it? Anybody? Do you know about chance discovery story by Fleming, Alexander Fleming? So penicillin was discovered by Alexander Fleming. So the whole story starts as in he was working on Staphylococcus. Okay, Staphylococcus. Hold on. He was working on Staphylococcus bacteria. Okay, Staphylococci bacteria. So you know what is Staphylococci? Must have taught you. It's a bunch shaped bacteria. The colonies, they appear in bunch shaped. So he observed that these particular bacteria were not able to grow in presence of a chemical. Okay, till then the antibiotic word was not given, not coined, so it was not able to grow in presence of chemical produced by penicillin notatum. So this penicillin notatum was what? This is a fungi which actually produces the antibiotic called as penicillin. Okay, so this is a fungi. Later onwards, we have two more people, Chain and Flore. Who made penicillin a full potential drug. Yes. So this is antibiotic part of the industrial products. Let me know if there is any doubt. Shillong, Tamilong, Itanagar. Yes, Shillong, just give me a thumbs up if things are clear. Not clear? And what are these three boys doing? No, on this side, on my right side. Okay, Itanagar, clear? Sorted? Confident of doing questions? Copied, Shillong? Okay, fine. Moving ahead. Tamilong, Itanagar, copied. Shall I move ahead? The third section of industrial products. We have chemicals from now let's see what chemical you are going to get from Aspergillus niger. So Aspergillus niger happens to be a fungus, and this produces citric acid. We have Acidobacter aceti, bacteria, species of bacteria which produces acetic acid. We have Clostridium butylicum, don't forget to underline them with species and genus. So it's binomial nomenclature. Okay. Clostridium botulicum, again a bacteria which produces butyric acid. And we have is lactobacillus. This again is a bacteria, and this produces lactic acid. Saccharomyces cerevisiae, this is a yeast. We all know yeast is what? Yeast is the fungus, isn't it? And this produces what? This produces ethanol. Just a minute, there is some technical issue. I have to rewrite one or two things. So this lactobacillus is a bacteria again, which is helpful in producing lactic acid. And so these are the particular products, these are the particular industrial products that we are using it, isn't it? We are using for our everyday purposes. So Aspergillus niger, this produces citric acid, it's a fungus, and Acetobacter aceti, acetic acid, butyric acid from Clostridium botulicum, lactic acid from lactobacillus, and ethanol from Saccharomyces cerevisiae. Fine? Sorted? Clear? Any doubts? No? No doubts? Fine? Clear? Okay. Tamilong, Itanagar, Shillong, everybody. So chemicals are done. Let's see enzymes from the microorganisms. So this would be what? This would be topic D. Fourth topic. Let's see enzymes. So enzymes, what kind of enzymes do you expect? Anybody? So detergents, they have lipases. You must have noticed the detergents that we use They are very helpful in cleaning the oil stains on the clothes. That is basically because of lipases. We have pectinase, and protease is used in bottle. Now, if you are making a juice at your place, isn't it, fresh fruit juice, that won't be that clear as compared to Tropicana or Real or any other brand. So this is why, because they are using these particular enzymes called as pectinase and protease. What they do is that they break down the cell wall component called as pectin as well as the protein sections. So when you clear out all those things, your juice will be very clear. So this is used in bottled juices. So next time you drink any bottled juice, remember it's clear because they have used enzyme called as pectinase. We have streptokinase. Now this streptokinase comes from Streptococcus bacterium. This is used as a clot buster. To remove clots. So many times coronary heart disease is because of formation of small clots. So what doctors do, they actually inject a particular enzyme called as streptokinase, which is obtained from Streptococcus bacteria, and this streptokinase goes in the blood vessels of the patient, and what they do is that they burst the clot. So all the clots are bursted when there will be the smooth flow of the blood in those vessels. So this is very important from the medical point of view. So we have three things to share under enzyme section, that is lipase, pectinase, protease, streptokinase. So three products of enzymatic importance. Fine. It's Tamilong, Itanagar, Shillong. Everybody clear? We'll move ahead with the bioactive molecules. So the fifth part of industrial products. So we have bioactive molecules like number 1, cyclosporine A, obtained from trichoderma from Trichoderma polysporum fungi, and this is used as, I have taught you this thing, immunosuppressive agent in those patients who have undergone organ transplantation. So cyclosporine A is the first bioactive molecule. Statins being the second bioactive molecule, obtained from a yeast called as Monascus. and this is used as blood cholesterol lowering agent. Fine. So we have these bioactive molecules as a component, as a part of our day-to-day life, where we are dependent on organ transplantation, we are dependent on the blood cholesterol lowering agents. For these products, we are seeing these microorganisms as a very, very valuable source, as a very useful source, and thus they are of great human welfare, uses to human welfare. Clear? Yes, Shillong, Itanagar, Tamilong, copied. By the way, what are you people doing on first bench right side? These three boys, what are you doing? What are you doing? What is going on? Nothing? Sure? Are you sure? Doesn't seem to be. Okay. It might be, but it is not, does not seem to be from here. Okay. Fine. Copied up to this, everybody? So are you able to relate these microorganisms in human welfare from your day-to-day activity? Are you able to? Yes. Fine. Very good. Okay. So can we take a small break? Oh, yes. Who is it? Edne? Edne is dancing. Is it Edne? I can notice everything from here. It doesn't seem to you, however, but we teachers, we notice everything. Okay, fine. So we'll have a small 5 minutes break. Quickly. It was too much of theory today, I know. But this chapter is actually very, very theoretical. I cannot help it. Really sorry about it. But this chapter is actually heavy. But yes, do remember whatever I am giving you in notes, please do not escape those points, because questions might come, but these are the basic things which will be asked. So don't study this chapter in very, very elaborate or under whatever I have taught you. Just be apt to this particular point, these particular points. We will expect questions from these topics only. Okay? Fine. Take a 5-minute break. We will back by 39, 40, 45. Yes, children. Shall we resume? I guess we should, isn't it? Okay, fine. So, won't be taking you now to a new thing. We'll just revise and rewind the last things which we, all the last things which we did. Fine with you? We'll take one more class, almost one class, 1 hour more, 1 or 1.5 hour more to complete this chapter. We'll do some questions again on the next class. So we'll just see the revision section from these topics. So whatever we did today. So microbes in human welfare, what we did was important components, isn't it? Microbes, we found them to be important components, and these important components are almost present everywhere. Okay? Then we talked about those places also where life actually seems to be impossible, like layers of snow deep inside soil. All these parts, they actually seem to be somewhat impossible, but yes, we do have life at those places also. In fact, acidophiles, thermophiles, psychrophiles were the typical terms that we talked about. And then microbes, we will talk about microbes in terms of bacteria and fungi. I told you the reason why, because they are grown on nutritive media, so they are thought to be more useful compared to any of the other microorganisms. So they are forming colonies and we are using them to study microorganisms. So household product, curd from milk. I told you the highlights why we prefer curd over milk, because number 1, it is partially digested protein, number 2, it is rich in vitamin B12 content, and number 3, it checks the disease causing microorganisms also in our system. And we talked about dough of idli and dosa, which again can be fermented using a baker's yeast called as Saccharomyces cerevisiae. Traditional drinks like toddy, again which is used in South India, is from fermented sap of palm plants. Fish, soybean, bamboo shoots, all are micro fermented products. We have cheese, different varieties. We talked about Swiss cheese, we talked about Roquefort cheese. Swiss cheese, large holes because of the yeast called as Propionibacterium shermanae, and Roquefort cheese is by typical fungi that is giving it a typical flavor, specific flavor. Do we have any doubts up till now? Shillong, Tamenglong, Itanagar? Any doubts? Yes, no? Similarly, we have the usage of microbes in the industrial products also, where we use them to synthesize a number of products including beverages and antibiotics. So we talk about these particular product formation, we need a big size vessel, large vessel, and those large vessels are called as fermenters. So first of all, we talked about fermented beverages, that is wine and beer, whiskey, brandy, rum. I told you the process, these are having lower alcohol and these have high alcohol because of distillation process carried on. But however, both of them, or all of them in fact, they are produced by a yeast called as Saccharomyces cerevisiae. So if you use Saccharomyces cerevisiae to produce CO2, it would be called as baker's yeast, and if you use it to produce alcohol, it will be called as brewer's yeast. So why brewer's yeast? Because they produce ethanol when grown on malted cereals and fruit juices. We have antibiotics, chemicals. Okay, I was supposedly going to tell you the story behind the first antibiotic discovery. Why we call them, why we call it to be chance discovery. The story moves ahead as in Alexander Fleming, he was working on this bacteria Staphylococcus. So he was growing all these bacteria on culture plates and keeping those culture plates aside. Every time he used to culture, he used to keep them aside, and he used to have an attendant who used to help him with the cleaning of these plates. So whenever he wanted new plates, he used to like ask his attendant to give him new plates by cleaning those old used plates. So once upon a time his attendant was not well, he did not turn up, and Alexander Fleming was in great need of those plates. So what he decided to clean those plates himself to continue that day's work. So when he went to do that, what he found that there were colonies of staphylococci growing, but somewhere the colony was not able to grow. So if this was particular space, this is space, the yellow one I will be marking, or in purple let me mark. This space was the area where staphylococci did not grow, this particular area. So apart from this area, everywhere staphylococci colony could be seen, apart from the purple thing. So this must be something, this particular colony must be something. When he went ahead and performed study, what he found that this was nothing but this was Penicillium notatum growing over there, which was releasing certain antibiotics, and those antibiotics inhibited the growth of the staphylococci colony in nearby areas, and thus was the first antibiotic discovered, which was called as penicillin. It was first antibiotic, and that's why we call it to be chance discovery. Okay, so this is chemical produced, and this Penicillium notatum is the fungi that produces this particular chemical. So later onwards he just discovered it, and he used a story, he completed the part from his side, and Chain and Florey were the next two people who actually used penicillin in a full potential drug, and this was a very turning point in the history of medical science, because in the First World War there were so many people dying, but this discovery, penicillin, was discovered somewhat near the Second World War, and because of this drug, this antibiotic discovered at that time, a lot many lives were able to save. We were able to save a lot many lives because this penicillin was available at the time of Second World War. So this was a very turning point for medical science. Now we don't use penicillin. Penicillin comes under first generation of antibiotics. We don't use it. Currently we are into third generation antibiotics because of antibiotic resistance, isn't it? We have done that particular part in evolution as well, that antibiotic resistance, how it developed among the microorganism population, and penicillin is a very good example. Now if you induce penicillin into somebody's body, it's very likely that they are going to develop penicillin allergy. So this is the next thing what penicillin is doing to our system. They might develop penicillin allergy. Fine. So this chemicals, if you talk about, we have citric acid, Aspergillus niger, acetic acid, Acetobacter aceti, butyric acid, Clostridium botulicum, lactic acid, Lactobacillus, and ethanol from Saccharomyces cerevisiae. Similarly, we do have enzymes, that is lipase, pectinase, protease, streptokinase, which are again very useful in their typical usage, that is laundry detergent, using making the bottle juices clear, and clot buster. Similarly, we do have bioactive molecules. What are the bioactive molecules? They are cyclosporine A and statins. Cyclosporine A, which is produced from Trichoderma polysporum fungi, and statins produced from Monascus purpureus yeast, which is used to lower the blood cholesterol level. And this is Trichoderma, which is used for immunosuppressive. Now, why would anybody use immunosuppressive drug? Anybody? Why would anybody use immunosuppressive drug? Or when would anybody use an immunosuppressive drug? Suppose you were a doctor, when would you give your patient an immunosuppressive drug? Yes, Itanagar, Tamenlong, Shillong, tell me please. What is a foreign substance according to body? What is a foreign substance? What is a foreign substance? Yes, very good. So what we do is that we use immunosuppressive agents or drugs to lower the activity of our T cells, because if immediately the graft has been implanted in somebody's body, okay, so that graft can be rejected. That graft is actually a kind of cell or a tissue which is not of body self. So our body has an immune system which actually recognizes self and non-self. This is my own, this is not my own. So whatever is not from my own source, I will try to remove it out. So what we do is that in that case, we use immunosuppressants like cyclosporin A to suppress our immune system from attacking the newly transplanted organ. So in this way, we are able to escape the graft rejection problems, but however, the patient depends on these drugs for a very long time, might be lifelong. Fine. Okay, I guess this must be enough for today. We will talk about microbes in sewage treatment part and the other sections in the next class. Go back home and try to learn. We talked about many new terms today, many new microorganisms today. That is why I decided to keep it a bit lighter. So just go back home and try to learn all the names by next class. I will be asking you questions in the next class, all the centers. Yes, Shillong, Tamenlong, Itanagar, come prepared for a small test in the next class. Yes, hit it hard. Harder. Okay, fine. Okay, fine then. Thank you so much. Yes. Just don't hurt yourself in this process. Be sure. Okay, fine. Okay. Itanagar, Shillong, Tamenlong, everybody. Take care. Bye-bye. Thank you so much. See you on Monday, next class.